Hello everyone. Welcome to our second webinar from the PAPERI webinar series. I'm Sarah Youssef, the PAPERI Education Working Group Coordinator, and I will be moderating today's session entitled Genomics of ALL in the Childhood Cancer and Leukemia International Consortium. I'm delighted to introduce our esteemed speaker, Professor Dr. Logan Spector. Dr. Spector is a professor and director of the Division of Epidemiology and Clinical Research in the Department of Pediatrics at University of Minnesota. He is a pediatric cancer epidemiologist who specializes in the etiology of leukemia, bone, and liver tumors. Dr. Spector has been a PI or a co-PI for 9 NCI-funded studies, and he's the immediate past chair of the epidemiology within the Children Oncology Group and the current chair of the Childhood Cancer and Leukemia
وبابا هنا ولا مش موجود برضه؟
International Consortium. I'm sorry for that. In his work, Professor Dr. Spector uses surveillance data, traditional case control studies that examines environmental and lifestyle risk factors and combine them with the genetic epidemiologic approaches. In these approaches, Dr. Spector mainly utilizes both single nucleotide variant array and next-generation sequencing data in order to understand the germline basis of childhood cancer risk. He utilizes different modes of analysis, including genome-wide association studies, transcriptome-wide association studies, gene burden analysis, de novo mutation analysis, and admixture mapping. The ultimate goal of Dr. Spector's research is to enable the prediction, early detection, and consequently the prevention of childhood cancer. In today's talk, Dr. Spector will provide insights on the genomics of ALL in childhood cancer and leukemia international consortium, and we will provide a brief on the Admiral Study and GWAS data of the African American kids with BLL. So please join me in welcoming Dr. Logan Spector. We will be having a question and answer session after Dr. Logan's talk. So I would ask you currently to mute your microphone, and Dr. Logan, the floor is all yours.
Thank you. Thank you, Sarah, and also thank you, Wafa, for inviting me.
Yeah, thanks.
Well, you gave a very nice introduction and the title, so I will skip that. I know that you got a lecture last week or the week before from my close colleague, Dr. Joe Wiemels. So there may be some repetitive material here, but this will give you another view, and I do have some very new data that should be coming out in the next few months from my Admiral study that you may find of deep interest. So I'll talk about the descriptive epidemiology of childhood cancer, the history of CLIC, and then tell you about the CLIC Genomics Project and Admiral. So this is a US-centric view of childhood cancer. Apologize, usually I'm giving lectures to people in the United States, but it is the #1 cause of death due to disease. In the US, it's estimated 1 in 285 children will be diagnosed with cancer by their 20th birthday. There are almost 16,000 new cases diagnosed each year in the US, and 40,000 children in active treatment. Globally, there are about 400,000 children diagnosed with cancer each year. And in the US, 20% of children with cancer will not survive it. I certainly understand that in low and middle income countries that the mortality rate is much higher. And I'm very dedicated to changing that statistic. So pediatric cancer, as you may know, is a small part of the overall cancer burden. These data are from the United States and show that cancer in people under 20, the National Cancer Institute defines childhood as 0 to 19, constitutes about 1% of the childhood cancer burden. And even though that's rare, It's even more rare once you realize that cancer in children consists of a heterogeneous group of tumors, which consist of several types that are only really seen in children, such as embryonal tumors, and other types such as leukemias and lymphomas, which are comprise over a third of childhood cancer, which is a much higher proportion than in adults. Also, sarcomas and CNS tumors are far more common as a proportion of cancer in children than they are in adults. In adults, the predominant form of cancer is carcinoma, which is a very minor type of tumor in children. So even within that heterogeneous group of tumors defined by site and histology, there is even more heterogeneity, which we've really discovered in the last 10, 15 years, defined by molecular genetics of the tumor. So here you see molecular genetics by age at diagnosis in acute lymphoblastic leukemia. And you can see that it varies wildly during childhood and consists of many different molecularly defined subtypes. A few other facts about childhood cancer. There is a distinct sex bias with males having a higher rate of childhood cancer than females for nearly every cancer. In some cases, it's very profound, as in Burkitt lymphoma, or fairly subtle, as in astrocytoma. There are a few cancers, including some germ cell tumors, thyroid cancer, and melanoma, in which females have the higher rate. And in the US, there are some interesting and persistent differences in risk by race ethnicity as we define it in the US. So for acute lymphoblastic leukemia, white children and Hispanic children have the highest risk, followed by a slightly lower risk in Asian children, but a profoundly lower risk in African American children. And we see patterns that are quite prominent and persist over years. For instance, this one in astrocytoma in the US, which also, by the way, mirror international trends. So we think that pediatric leukemia and many other childhood cancers start in utero. There are quite a few natural history studies in twins and using cord blood and dried blood spots to backtrack leukemia to birth. And then that has focused a lot of the epidemiology, risk factor-based epidemiology on gestation as a critical window of risk for leukemia. So I have really dedicated my career to understanding why kids get cancer in order to enable risk prediction, early detection, and someday prevention. And several years ago, my colleague and successor as chair of epidemiology for the Children's Oncology Group, Philip Lupo, and I did wrote a review of risk factors for childhood cancer. This heat map is not systematic or quantitative. It was merely put together by our opinion as two experts in the field. But you can see for ALL, there are a lot of risk factors that we think increase the risk, and we've divided them into those that operate during the preconceptional period or pregnancy, around the time of birth, and then there is a role for risk factors during childhood as well. And if you concentrate on the odds ratios here. There aren't a lot of very strong ones. In fact, usually epidemiologists would say a relative risk of 2 is strong. But in the context of pediatric cancer, a 1.5 or its inverse, 0.67 would be considered fairly strong. There aren't a lot of strong environmental risk factors. So CLIC researchers are located all over the world. As of June 2024, we had studies on 6 continents, and I would like to thank WAFA for being our foothold on the African continent. I hope that talking to your group will spark collaborations with other areas of Africa, in particular sub-Saharan Africa. We have 19 countries, 67 principal investigators or delegates who hold 50 studies. As best we can tell, because we're still, even after all these years, trying to get a hold on our data, we have about 48,000 leukemia cases, 11,000 CNS tumors, 23,000 non-CNS embryonal tumors, and largely because of the participation of registry-based studies in the Nordic countries, nearly 4 million controls. We have 1 data coordination center, which is at IARC in Lyon. And I saw Felix on the call. I hope you're doing well, Felix. So here are our sites. I mentioned that Egypt, the Egyptian study is our foothold in Africa. And but I see there are other missing areas. As you can see, we are heavily invested in Europe and North America. We do have a couple of studies in Central America, and then 2 in Brazil, 2 in East Asia, and then Australia, New Zealand. But it is my ambition to expand to areas where we are lacking. Sub-Saharan Africa is a large one. Most of the rest of the continent of South America, there is still a need for more studies from Central and East Asia, and we have very little representation from South Asia, i.e. India or Pakistan. So CLIC was founded in 2007 by the late Pat Buffler at the University of California, Berkeley. In 2013, we had our first publication. And also Dr. Buffler passed away, at which point Catherine Métayer, who also is at UC Berkeley, took over as chair. In 2014, the Data Coordination Center was established at IARC in Lyon. In 2019, CLIC voted to open to childhood solid tumors as well as family designs. Previously, CLIC stood for the Childhood Leukemia International Consortium, but if the rationale for pooling leukemias was the rarity, there was even stronger rationale for solid tumors, which are individually much more rare. In 2020, we established the CLIC Foundation as a nonprofit in the United States to help raise funds for CLIC. And in 2022, we had a ALL genomics grant funded by the U.S. National Cancer Institute. So our governance is this. I'm the chair for the time being. And I have a management group consisting of Dr. Joachim Schuss of IARC, Julia Heck, who is now at the University of North Texas, Dr. Michael Schurer, who's at Baylor College of Medicine, and Dr. Maria Pombo de Oliveira, who is an emerita investigator at the Brazilian National Cancer Institute. We have a coordination group which functions as the voting body of CLIC and consists of the PIs of member studies. And then in the last year, I hired Iris partially as program manager for Click. So she helps keep everything together. Currently, our model is that we have concept proposals that member studies agree to participate, and then the member studies provide an analytic data set to the proposing PI directly, although the data coordinating center may provide core variables to the proposing PI, and then the analysis takes place at the proposing PI's institution. We are in the process of trying to centralize data because the previous process was very slow. So a concept proposal would go to the members who could agree to participate, but we're trying to move entire data sets to the data coordinating center, which would provide an analytic data set for use on the IARC scientific IT platform, which means that somebody will remote in to data that stays at IARC. So I would just like to talk about some of our greatest hits over the years in the non-genetic epidemiology, which you may nevertheless find interesting. Years ago, I was asked to lead a pooled analysis, and I chose as the topic mode of delivery because I thought that there are really only two ways into this earth, and it would be a very easy variable to pool. Of course, once we got into it, it turns out that it was more complex than you might imagine. So we thought that because microbiome is different depending on the mode of birth, cesarean or traditional, that would be a driving factor in risk of childhood leukemia. But what we found was no association of ALL with mode of birth. But if we looked at pre-labor C-section, there was an increased risk, which was small but statistically significant. And we think what this means is we know that there are circulating pre-leukemic clones in some children, and also that cortisol is higher during labor. Cortisol is mirrored by glucocorticoids, which are basically synthetic cortisol and are a backbone of ALL therapy. So our thought is that perhaps the cortisol during labor has an effect of culling out pre-leukemia, and we picked that up in an epidemiologic study. We also, there are longstanding hypotheses about immune stimulating exposures or, early exposure to infection being protective for ALL. And in our analysis from a few years ago, we took the largest look at this question. We found that the longer a child breastfed, the lower their risk of ALL. We did not find a lot of association with birth order, which can be a proxy for infection exposure, but we did find a inverse association with daycare in the United States, which is commonly held to markedly increase a child's exposure to infections. And this actually was dose dependent. So the earlier a child started in daycare, the greater the degree of protection. We also have published our first paper on a solid tumor. It was on breastfeeding and risk of childhood brain tumors, which was in fact almost completely null. And then this paper is currently nearing submission. We decided to look at smoking and childhood cancer across types. But because we don't think that we get good data from interviews, we concentrated on registry-based studies, which eliminated both selection bias because these were 100% samples of the areas in which we looked, and also recall bias because these data were collected prospectively. And we also, because that left measurement error as the only big source of error, we did a quantitative error correction where we looked up values of sensitivity and specificity for maternally reported smoking and corrected for it. And what we found was that whether we looked at the bias adjusted or non-bias adjusted values, they were almost the same and almost universally null. So although it's counterintuitive and not a great public health message, it does not appear that maternal smoking during pregnancy is a major cause of childhood cancer. We also have looked at parental age, and here we see that there's a good example of bias, because in our case control studies requiring interview and participation, we saw no association of parental age with childhood leukemia, but in the registry-based studies in which selection bias is eliminated, we did see an increase. All right, so moving on to the topic under study, or topic of your consortium, which is genetic risk. It has long been known that ALL is highly polygenic with stronger per allele odds ratios than in adults. This is from the 2019 GWAS for BALL, which was mainly in children of European descent. And here we have risk ratios along the polygenic risk continuum, and this is measured from the median. All right, so in the highest percentile compared to the median, it's nearly a 5-fold risk. If we compared to the lowest percentiles, it's actually more like 10 or 11-fold risk. And this was with SNPs that were known in 2019. For those of you who follow this literature, by comparison with a adult tumor like prostate cancer or breast cancer, they have 150 SNPs. Let me back up. This was about 15 SNPs contributing to this polygenic score. In breast cancer and prostate cancer, it's more like 150 SNPs, which from the bottom to the top percentile increase risk only about 5-fold. So that should give you some idea of the much higher degree of association of SNPs with ALL. Increasingly, well, first of all, the National Cancer Institute, as well as many investigators, are crucially aware that it is not ideal that most GWAS have started in people of European descent. And there is a massive interest in diversifying GWAS in order to increase sample size, but also to see if there is population-specific associations. Our colleagues, and you might have seen this last week, because this is Adam DeSmith, who works with Joe Wiemels at University of Southern California, but they took advantage of the diversity in California to focus in on risk in Latino children, who again have the highest risk in the United States. And although the IKZF1 locus, also known as Icaros, is long known to be associated with ALL, and that was discovered in European children. Here they found in red a variant which appears to be specific to admixed Americans, or Latinos, as we say. And it's an allele that appears to have risen up in South America thousands of years ago, and must have had some selective advantage because it became more common in these children. It became more common in the population over the years. And it appears to increase risk to such a degree that it may contribute to the higher risk in Hispanic children. Okay, so now I'll discuss early results of my Admiral study. And I'll tell you what that stands for. But the main impetus for this study was the observation that in both North America and in sub-Saharan Africa, there is a lower rate of B-ALL in Black children. And previously, there had been a thought that perhaps these children are not coming to clinical attention in low-income countries, but there are IARC certified gold standard cancer population-based cancer registries in sub-Saharan Africa now, which confirm that the lower rate seems to be organic. I also looked up rates in Jamaica, which is primarily populated by the African members of the African diaspora. And you can see that the rate is very similar to the rate of Black children in America. So I think that that these lower rates are organic and not artifactual due to under-ascertainment. In the US, if you ask a pediatric oncologist, they will all tell you that T-cell ALL is more common in Black children. And that is true as a proportion of ALL. But once we put on denominators, it's very clear that T-cell ALL has the same incidence in Black and white children in the US, and there is simply a lower risk of B-cell ALL in Black children compared to white. So it's not that T-cell ALL is more common in Black children, it's that B-cell ALL is much less so. We looked at data from the SEER program, which made a file available to look at, to estimate socioeconomic status. It was based on address, so not a perfect proxy, but in the US, as in almost everywhere, where you live does tell you something about people's SES. And we were looking at the ratio of incidence in white to non-white within strata of socioeconomic position, as we said in the paper. And interestingly, when we were comparing the rate in Black and white children, there was a deficit or a difference, a lower rate in Black children at all strata of SES, suggesting that SES differences, which are well known in the US, did not explain this. We also looked at higher confidence risk factors for childhood cancer, ALL, in African American and white children. So these odds ratios are for meta-analyses for pesticides, residential traffic, petroleum products, solvents, and some protective factors like breastfeeding, daycare attendance, folic acid supplements. And then we looked up difference in exposure. And for most of these exposures, the risk factor, which increases the risk of ALL, fell more heavily in Black children. The only exception was daycare attendance. Black children were more likely to attend daycare than white children in the US. Lastly, these data were from a record linkage study, which I conducted many years ago. And on birth certificates in the US, the parents' race and ethnicity is listed. And because of the large size of our study, we were able to look at birth certificates where both parents were listed as white, both parents were listed as Black, and those where one parent was listed as white and one as Black. And what we see is that the relative risk of ALL, although the confidence interval is wide, was directly in between that for 2 white and 2 Black parents. Of course, it is not easy to say that environment would be exactly in between, but genetics would be by definition. So on the basis of these observations, I started the Admiral Study. That stands for the admixture analysis of ALL in African American children. We get cases from the Children's Oncology Group, from institutional biobanks, and 2 population-based studies in Michigan and California. Our goal is about 1,500 Black children and 600 controls. We use the global diversity array, which has been shown to have much better coverage and provide better basis for imputation in Black children or Black people in general. And our aim 1 is admixture mapping to and GWAS for etiology, which is what I'm about to show you. And then aim 2 is admixture mapping for outcomes, because there is some evidence that Black children more often have a failure of induction as measured by minimal residual disease, and there is documented lower survival. So first we looked at global ancestry in SMILES, which is my statewide Michigan leukemia epidemiology study, so perfectly population-based. We found that cases were, on the whole, about 8%, had about 8% less African content to their genome compared to controls, and this was very significant. In other cases collected on a nationwide basis and other controls also collected on a nationwide basis, there was a smaller but still very significant lower global African content in cases compared to controls. In the CCRLP, which is the California Childhood Record Linkage Project, it was actually the same exact global ancestry estimate, and we're still working out whether that was due to some study design choice. But overall, there appears to be global patterns are reflecting our hypothesis, which is that there is some genetic factor or factors which combine to lower the risk of BALL among children of African descent. When we looked at the SMILES, the Michigan data by age of diagnosis, the effect was present in all ages, but seemed to be higher at older ages of diagnosis. All right, so moving on to the main findings, we have completed a GWAS, genome-wide association study in the Admiral Study, and 2 loci in blue came out as highly statistically significant. These are the IKZF1 and ARID5B, which were the first 2 loci described as to be associated with ALL in European children. And so this is a very robust finding of transethnic risk for ALL. But in red, we have several genes which came out of the analysis, which are completely novel and which are seen only in populations with large African genome content. And so I apologize for the busyness of this slide, but it is what it is. You see that we have discovery in COG cases where these loci have very strong per allele odds ratios. So 2, 2.3, almost 3 here. By contrast, in say prostate cancer, the top hit has an odds ratio of about 1.2. We saw very good replication, and then in meta-analyses, these are very strongly significant on a genome-wide basis. What is truly interesting is that the allele frequency is the alleles are almost absent in other populations. These are African-specific alleles or specific to African segments. They are present in African genomes and 1,000 genomes as the superfamily at a low prevalence. And there is a little bit of detectable prevalence in AMR, which is admixed Americans, or what we'd say is Latinos in the United States. 80% of Latinos in the US are specifically Mexican. It is perhaps not well known, but there was chattel slavery in Mexico. And as a result, there is a small proportion of Mexican genomes which are detectably derived from African ancestors. So my suspicion is that this low prevalence of these SNPs in Mexicans or Latinos is due to the African ancestors. When we put these together as polygenic risk scores, we get a much higher relationship using the African-only SNPs as opposed to the SNPs that were just the transethnic SNPs. And this is perhaps not surprising because while the per allele odds ratio in the transethnic SNPs are strong, around 1.5 to 1.7, they're not as strong as the African-only SNPs, which again were a minimum of 2 and went as much as 3 per allele. So that now Admiral Study is now feeding into the ALL genomics project, which is pooling childhood ALL GWAS data sets from around the world in order to take advantage of the genomic diversity which is present in click studies. Our first aim is to estimate heritability of ALL using SNP-based methods in diverse populations. Aim 2 is basically to create a comprehensive genetic architecture for ALL, and aim 3 is to create population-specific polygenic risk scores and examine the relationship to harmonized epidemiologic data within CLIC. Here's our funding. And again, I apologize for the table being busy, but this tells you, what years the studies were, what our age groups, age diagnosis are, and then the ancestries will have we expect far more children of African or African American ancestry because this was the numbers present in 3 years ago when I wrote this, and we're already exceeding it with the addition of Admiral. But we obviously have a lot of European children, but substantial numbers for admixed Americans, East Asian. Thank you to WAFA for maintaining the Egyptian study. We also have a large number of Middle East and North Africans. The two areas with 1,000 genome superpopulations where we are under ascertained children are South Asian and Southeast Asian, and we're very dedicated to finding data sets that will add to those numbers. Because of the era of the studies, we don't have complete tumor phenotyping, but we do have substantial numbers of children with some subtypes of childhood ALL. And at the bottom, you can see what kind of epidemiologic data we have. It's working out where click studies have about 12,000 children with genomes, and about 2/3 of those are attached to robust epidemiologic data. So we have a little bit of a complicated data structure, and that's because of laws in Europe, the General Data Practices Regulation, as well as a law in California, which governs the use of data derived from newborn dried blood spots. The University of Southern California, Joe Wiemels, is the Analysis Center for the Americas. INSERM in Paris is the Analysis Center for Europe and Africa. And then the University of Newcastle in Australia is the Analysis Center for Asia Pacific. And the University of Minnesota is the coordinating center. We write programs which we push out to the analysis centers. They do the analysis and they send us summary statistics, which we then combine by meta-analysis to make our insights. Click member studies also have data from, data in waiting, meaning DNA samples, which I am working to try and turn into genotypes. Actually, Mexico, working with Adam DeSmith at California, has already been genotyped. And I'm currently working with Denmark, trying to get those genotyped. But then there are very valuable collections, particularly in India, which, as I just said, was under ascertained, and in Brazil, which is even more of a melting pot than the US. And I will note, has the largest African diaspora population of any nation, more so than the United States. And so we are trying to grow the diversity of these of our samples. So when we're pooling is done, we should end up with about 40,000 childhood cancers with array genotypes, about 20,000 ALL. As I mentioned, 12,000 ALL in click studies. We are getting public data as well to add to the resource, and about 8,000 ALL with questionnaire data. If you're interested in lymphoma, we have about 3,500, about half of genotypes. And this slide was actually for an earlier lecture of the Interlymph Consortium, who study adult leukemia or adult lymphoma and leukemia. Just wondering what relationship that has to pediatric leukemia. And there is a generally increased risk of hematopoietic cancers in those with childhood leukemia and in their relatives. So with this resource, we have many analyses that we can do. We are talking about a pan-cancer GWAS, so looking for cancer pleiotropic genes, looking at copy number variants, somatic mosaicism, aneuploidies, and uniparental disomy, taking advantage of the genomic diversity to do transethnic find mapping, which basically uses the difference in haplotype structure between populations to help narrow down the list of causal SNPs rather than just associated SNPs. Mendelian randomization in order to assess causality, multi-ethnic transcriptome-wide association study, GWAS by single year of age, which could be of interest, sex-specific analyses, which could help explain the male excess of ALL. One of our aims is already maximizing risk prediction. And then finally, gene by environment interaction. I want to just tell you too about an effort that we have. This grant is under consideration right now. It's called maternal. The proposal is to genotype parents, but specific, and analyze mothers in click studies where the children are already genotyped. Our aim one is to conduct agnostic scans for variants or genetically determined traits among mothers of children who developed ALL. Two, to conduct focused analyses of maternal traits that are known from the epi to modify risk of ALL, such as obesity, preeclampsia, diabetes, gestational length, and birth weight. And then aim three is to conduct a first of its kind integrative analysis of breast milk genetics with offspring ALL risk. Here's the team. And I would say concentrate on the lower part here. We think we will end up with about 3,400 child genotypes, from which we'll have 3,300 maternal genotypes and 1,600 paternal genotypes, and then 2,200 controls, of which almost 2,200 will have maternal genotypes. It is, because of the location, largely European, but about a third of the cases will be Latino. And these studies have very good tumor phenotyping, so we'll be able to look in subtypes. So, we are looking to do a lot of future research, including genetic overlap of pediatric with adult heme cancer, maternal genetics, very interested in HLA and KIR genotypes, which are mostly ignored in GWAS because these are very complex regions, but they are highly relevant to an immune-related cancer. And then looking to do what we call integrative studies of perinatal genetics, integrating those studies into our GWAS. So I am happy to take questions now, but if you think of something later, please feel free to email me. My email address is spector@umn.edu. And now I will pause for questions.
Yeah, thank you.
Actually,
Thank
yeah.
you, Dr. Logan, for the presentation. It's very insightful. And for all of you who have questions, I appreciate that you send it on the text box or just use the raise hand sign. So, and I can see, just to mention that researchers from Nigeria and Tunisia are writing in the chat, they are very interested to collaborate. So I think they will be contacting you by email.
Please do.
Yeah.
Thank you, Nigeria. I see that Faisal had a question. How do variations in healthcare systems, practices, and cultural contexts across different countries impact the data collection and interpretation in international research? And what strategies do you and your team employ to ensure generalizability of findings? WeClick is not really in the business of starting studies as opposed to pooling existing studies. So I don't personally and have not invested a lot of time in understanding, what the barriers and challenges are in each area. I can tell you what they are in the US in very fine detail. As far as the generalizability of findings, having studies from diverse countries and economic kind of statuses is very important. We do sometimes stratify our analyses into the record linkage projects and the participation-based studies, because selection bias can affect the participation-based studies. And then as far as generalizability, I am very interested in cultivating studies and click membership from low and middle income countries, because I understand that the exposome will be different in different areas. So, you know, the main answer is to be cognizant of study design and the potential biases. And then secondly, to have studies from diverse regions and diverse genomes. There was another question. What are the key ethical and privacy challenges associated with handling large-scale genomic data and research? And what measures can be taken to address concerns related to patient consent, data security, and potential misuse of genetic information? Great question. First, we you know, make sure that all patients are consented and that our use of the data respects the consent that patients signed or affirmed. And secondly, we hold the data at genomic centers which have very strong data safety security. And thirdly, only vetted researchers who sign confidentiality pledges are allowed to use it, and they are made to agree not to try and re-identify anybody. And then thirdly, we don't send the genomics to individual investigators. Rather, they either will need to remote in to use the data in a walled garden, or the analysis will be done by the analysis centers. You know, the main, because of this issue, there is a very robust ecology and set of analyses you can do using summary statistics. And I highly encourage those on this call to become familiar with those, because the summary statistics cannot reveal an individual and are very widely available. And if you're able to use the summary statistics rather than the original genomic data, you will be able to contribute to this area of research much faster than if you need to obtain the original data. Is there any collaboration between the International CLIC and the International Cancer Genome Consortium? To my knowledge, the ICGC concentrates on somatic genomes, and we concentrate on germline. So we haven't yet collaborated. Let me pause for to take a question from Austin, who has his hand up. Come here, buddy. Come say hi. Sorry, it's morning here, and my son wants to say hi to everybody.
Yeah.
Wafazmet. This is my son, Dylan.
Hi, Dylan. Nice to have you. How are
Can you
you?
wave to everybody?
Hi.
All right. I have to keep talking. Okay. Bye-bye. Austin, you had a question?
Austin, please unmute.
All right, thank you for giving me an opportunity to ask a question. I've listened very carefully to the presentation. My question is, I just wanted to know if there are guiding principles on whether if one wants to share genomic data between the continents, across the continents, do we have the principles, the measures or some procedures that one need to follow when one wants to share the genomic data across the continents?
Yeah.
Let's say from Africa to...
All right. We spent a very long time coming up with a data transfer agreement, which would protect the interests of the participants and the PIs who generated the data, as well as CLIC. You know, the data would need to be sent from Africa would need to be sent to Paris, but it would have very strong protections, both privacy protections, but also protections of the rights of the PI. And one of the principles that is true for all our data in CLIC is that there's no use of the data for an individual study without the PI's approval. So, right now I have approval to do one set of analyses with the data that's being pooled. And, if I come up with another idea, that needs to be written up and circulated to the PIs. And then as far as and they need to approve each instance of use. And then if a investigator who is not part of CLIC wishes to work with the data, we can work on that. Eventually, I hope to migrate all the genomic data to IARC as well. And then we will have a very workable system where people can remote in the IARC scientific IT platform, and their analytic data set would be broken off for them to work with. So thank you for your question. I see a hand up from Jorge. You're muted.
Hi, Dr. Logan. Thank you so much for the talk. Really insightful. I have a question related to along lines of treatment. I might be wrong on this, but can you use transplants like bone marrow transplants for ALL? And if you do, would the genotype of the donor matter to the outcome of the treatment?
Yeah, well, transplant is a rescue treatment for ALL. It can be a frontline treatment for some forms of AML. And so it is, it is used in ALL, but it's a very small proportion of total patients, and usually after multiple relapses. And the second part of your question was then, can I use those patients in a GWAS?
Oh no, would the genotype of the donor who's donating the transplant matter, given there could be genetic
Yeah, that's been investigated by colleagues in the US. If you look up Teresa Hahn, Hahn, and Lara Sucheston Campbell, they have published on that very question. I actually published on mitochondrial genetics and of the donor and transplant outcome, and it was a null study. But the group, I think the study is called Discovery BMT. They've looked at that, but I off the top of my head, I can't remember their specific findings. But yes, there was some relationship of donor genotype to transplant success. And then more recently, this was in the news in PNAS, a colleague at University of Minnesota found that there was an effect of the donor's socioeconomic position with the transplant outcome, which is honestly mind-blowing, and that's probably why it was very hard for her to get it published. And presumably that's not a genetic effect, but epigenetic.
Wow, fascinating. Thank you.
It is. Where are you? And you sound like you're from the UK. Yeah.
Yes, hey, George from Welcome Connecting Science from UK.
Got it. All right. Well, I only have time for one more question here. It said, are there any correlations of CLIC findings between African American and African children? We have not yet completed the data cleaning and finalized the analytic data set for the CLIC genomics. So that's a great question. I do suspect we will have some, African immigrant children in the US, UK, French data sets, and maybe some other countries, but it won't be a large number. But that's a great question to ask. I also, we got my colleague at Baylor has a colleague in Uganda who has been collecting genomes or DNA from pediatric ALL patients in Uganda. And it's a very good question that we are pursuing. What is the connection between ALL genetics in African Americans who, the majority of their genome is derived from West Africa, but also on average 20% European derived, versus African genomes where there's no admixture, and of course a lot of diversity depending on where people are from, genomic diversity. So we're very keenly interested in that question, and we'll be getting to it in the coming years. But it's it's a lot of work. I'm going to put my email in the chat. As some people asked, but
Excuse
you're
me,
also
Dr.
well.
Logan. A standard
Yeah.
question we ask for our speakers for advice
Mhm.
for young researcher entering the field of epidemiology and genetic research. What is some skills they should gain, areas they should focus on?
What areas should they focus on?
Skills and areas, yes.
I think first of all, you want to start out with focused questions. Being as spread thin as I am is a privilege of having been in the field for well over 20 years. But when you're first getting your foothold in research, you need to narrow in and become a recognized expert in one small piece of the puzzle. I had a colleague who said, if you write 3 papers on a topic, you're an expert. So, pick a topic and get 3 papers and maybe more before you decide to branch out. That's general and not due to not any particular any particular field of disease. In terms of genomic research, I'll just say that the storage and computational power needed for using the original data is very large, and as well as the complication of moving data sets from one place to another. If you're interested in genomics and want to make a faster impact with less infrastructure, I really would suggest concentrating on summary statistics. Widely available, you don't need agreements to get them, and they contain a lot of useful information. I'd say thirdly, integrative studies, and let me just explain that if you don't know what it means. Think of a transcriptome-wide association study. There is some degree of gene expression which is under genetic control, and if you have a GWAS data set, SNPs that predict expression in a particular tissue, you can integrate those data sets and seeing gene expression. Powerful technique that because it collapses everything down to the gene level, which means fewer less of a statistical penalty, and also it's in some ways the only practical way to conduct. Going to have a data set where we directly measure gene expression in fetal livers, but there are data sets out there where they have SNPs and gene expression in fetal livers for fetuses that did not make it, and but we can attach that to our GWAS data, and it creates something much more powerful. So I would say for those of you in institutions which amplify the impact of your research.
Thank you.
Thank you so much, Dr. Logan. Yeah, I believe we
It's
have.
my pleasure.
It's our pleasure to have you today, indeed. So thank you everyone for attending, and thank you so much again, Dr. Logan, for this nice presentation. I would just like to ask every participant to fill in the post-webinar survey after the meeting ends, and I would like you all to have a nice day. Yeah, thank you again for attending. Bye.
Cheers. Have a nice day. Bye.
Yeah, you too. Bye.